Hi, everyone. My name is Asuz Daglar. I guess I should use this. So I'm a faculty member in EECS and IDSS, and I'm also the Associate Department Head of EECS. I'm very happy to introduce our next plenary speaker, Dr. Jennifer Listgarten. Dr. Listgarten is a senior researcher at Microsoft Research New England, located right next door. She received her PhD in computer science from the machine learning group in University of Toronto, and then joined MSR Redmond. Her area of expertise is developing and applications of statistical machine learning methods for the analysis of large-scale biological data, so computational biology, bioinformatics. And her recent projects focus on statistical genetics, immunoinformatics, and also CRISPR guide design. And I asked her, what is CRISPR guide design? She said, stay for my talk and you will learn. So with that, please join me in welcoming Dr. Listgarten.
Thank you very much. Great. Thank you for having me here. So I'm going to tell you a lot about genetics. And before I do that, I think most people here probably realize by now that genetics touches on many aspects in society. And I've just listed a few here. Precision medicine, you may not realize also agriculture to a large extent, and translational medicine, and finally basic biology, just trying to understand what's going on in living systems. So this is a pretty broad swath of things that it touches on. And so originally genetics was not a data science, but modern genetics is a data science. And the obvious reason for that is the existence of data. So here are some plots that if you are in genetics, you've seen many times. Basically shows you how the cost of DNA sequencing has gone down more rapidly than Moore's law. And consequently, the growth of DNA data has become just really, really massive. And so it is truly a data-driven science right now. So I'm going to also mention a little bit about epigenetics. Who here has heard of epigenetics? Wow, lots of people. Okay, great. So epigenetics are sort of chemical modifications that lie on top of the DNA, and they can alter its shape and how it sort of folds, which has implications for how it functions and which parts are accessible. And so it's increasingly being implicated in things like aging and cancer and these kinds of things. And here's an example just to show you how important it is. These are two genetically identical mice, but because they have different epigenetic profiles, one has a straight tail and the other one does not. So this is a sort of new era of genetics. Right, and so I'm also, at the end, I'm going to tell you about something that's been really hot off the press recently, which is how you can edit the genome as well. And this also has some really interesting applications, from correcting genetic disorders to, say, editing receptors in the body that could prevent you from getting things like HIV, and some kind of crazy sci-fi sounding things like gene drives. Who's heard of a gene drive? Right, so gene drives is a way to sort of impose a super inheritance of some trait artificially. And so you could actually wipe out something like malaria mosquitoes or something you might not want to wipe out. So it's a little scary. I don't work on that, but it's good to know about these. So what are some of the data science challenges in this sort of general area, and more broadly, I would say also in computational biology, which is kind of my broader area. So it's pretty large scale data, but the flip side of that is that it is large and unwieldy, but at the same time, it's very, very expensive to acquire enough data to actually make sense of it. And so you have this tension between having so much that it's unwieldy, but not quite enough that you can't do a lot in many cases. And sometimes that's because of combinatoric explosion or hidden confounding factors, which I'm going to tell you quite a bit about in just a second. And in reference to the previous speaker, there are, of course, issues of teasing apart causality from association. And so, my background is machine learning, and so a lot of the techniques I use focus on machine learning approaches, such as supervised, unsupervised, semi-supervised models, and also graphical models, which have been mentioned as Bayesian networks. But also in this area, it's actually important to leverage a lot of frequentist statistics to get traction with the communities who care about these things. So there's quite a large set of interesting tools and methods development problems in these areas. And so within the scope of that sort of overall theme, I'm going to tell you 3 stories of some of my recent work over the past 5 years. And the first is on finding the genetic underpinnings of disease. Then I'll do a really brief analogous story in the epigenetics world. And in the second half, I'm going to close with some tell you about gene editing. Right, so a genome-wide association study in its very, very simplest form is where we collect, like let's say people in this room are the cohort of my study, and I'm interested in some trait, maybe curly hair, although I think there's not enough people in here to do that, or some disease. And so I'm going to measure systematically your genetics. In the old days, that might have been 100 markers on the genome. Nowadays, it's in the millions, and these are called single nucleotide polymorphisms. And so I'm going to measure this massive list of genetics markers for everybody in the room, and I'm going to also have a trait label, and then I'm going to ask questions about how do the genetics affect that trait. And the very simplest question that I can ask here is to actually just scan through one of these markers at a time and say, is this associated with my trait? And as if you're at all a statistician, this will seem like the most sort of naive, trivial way to analyze things, and indeed it is, but people have actually spent millions of dollars analyzing things in this way, and they still do to this day. And I'm actually going to focus on that because even in that very simple setting, there are some difficult problems that arise, as well as in the more complicated ones. And I want to focus on those other aspects of modeling. And so everything I'm going to tell you about, just think that I'm scanning through the genome and I'm measuring one marker at a time, essentially getting a p-value, saying, is this implicated in my disease, yes or no? And so when I do that, if I make some sort of standard modeling assumptions, so technically IID, where I assume that the individuals in my cohort are identically and independently distributed, then in general, for this kind of data, I'm going to shoot myself in the foot. And so that's because there are several reasons that this is violated, and that includes people who are related to each other. So often we collect sort of pedigrees of families, grandparents, and siblings, and this violates that assumption. And then something that plays a really big role is actually ethnicities. So genetics vary by ethnicity, and this can completely mess up your analysis if you ignore it. And so basically, you can completely miss all the signal in your data that you've just spent millions of dollars in going through ethics approval to collect and so forth. All right, so let me give you a little bit of intuition about how that problem arises, and then some of the solutions and some of the challenges there. So this is already quite an old figure from a tutorial in 2006, and it shows what we call a case control study. So you could think of that as someone, a set of people who have cardiac disease and who don't. And sort of embedded in there are these hidden populations, and let's think of these as ethnicities. And the key thing to remember here is that ethnicity is a predictor of what marker you have at a given position. So I'm showing you one position here. It's an A or a T. And if I know your ethnicity, then I also know that you're that there's a probability distribution associated with your ethnicity and whether you have an A or a T, and it's different for different ethnicities. And so the implication of that is that if I come along and I ignore these populations and I just do a simple statistical test that says, are there a difference in the proportions of A's to T's between the cases and controls, then naively the answer is yes, that there are actually more T's here. However, if I know about these underlying populations, then in fact that's the true underlying explanation, and this locus or this marker is not actually associated with my disease. And so, and if you ignore this problem, and one, this problem is pervasive in almost every data set, and two, if you ignore it, you really mess up your analysis in a substantial way. And I've shown you sort of a simplified, it's not simplified, I mean, this happens, but it's discrete because it's case control. We also sometimes have quantitative traits like blood pressure. And it's also discrete, and then I'm showing you two very distinct populations, but you can back off of that and think of sort of smoothly varying populations. And I can generalize on both of those axes, and this problem still persists. All right, so what do we do? So first of all, we know we need to change our models and that don't make this assumption of IID-ness. But what's really important actually in this area is to have some idea of, are you using the right model? And the good news is that because we're doing massive scale hypothesis testing, we can get some insight into that. And so let me explain how we can do that. So imagine that I'm measuring something like 500,000 or 1 million of these markers, and each one I'm getting a p-value for its association with the disease. Then it's a reasonable assumption for most genetic traits that very few of those million markers are going to actually truly be associated with my trait, right? So, maybe it's 100, maybe it's 1,000, but it's some sort of small amount of the million. And what that means is that the bulk of my p-values should be drawn from a null distribution. And it's a simple null distribution that we know. It's simply uniform on 0, 1. And so what that means is, under that assumption, I can actually compute a p-value for every marker in my data set under some model. And this is actually minus log p-values, so these are highly significant. And so I take the ones from my data set that I've computed, and I plot it against the expected ones under this null, which is just uniform on 0, 1. And if it matches exactly on the diagonal and within the error bars, then I know both that basically I know my assumption is valid, that it's completely drawn from the null distribution. And of course, in a sense, that's not what we want, because we want to actually find signal. So when we find signal, it actually tends to do that and then peel away. But something that is systematically well outside of this envelope means that you've made an error in your modeling. And so this is like a really nice property that lets us help deduce whether we're doing good things in modeling. And in contrast, this is a model that I'm going to tell you about on the same data set. This is also a real data set, by the way. This is not a cartoon. This is what the new plot looks like. And in fact, in this data set, there is no signal. So, and I'm going to show you a lot of these. Are there any questions on this plot? Does that make sense? Okay. Right. So I've told you this intuition that what's going wrong here is essentially a lack of homogeneity in the relationships between people, either because of pedigrees or because of ethnicity. And so it's probably not going to surprise you terribly when I tell you that the starting point, therefore, of a solution to this problem is going to be to estimate that similarity and then incorporate it into a model. And so the sort of beast that we compute is this genetic similarity matrix. And I've shown you 3 examples here, so let me just walk you through the first one here. So this has 450 by 450, so imagine there's 450 people in this room. Shows you the genetic pairwise similarity between every 2 people. And this is an example where the data are IID, and you can see that because it's homogeneous on the off diagonal. And this is an example that has, and these are real also, by the way, these are not cartoons. This has pedigrees, so grandparents, siblings, and these kinds of things. You see this very fine-grained structure that specifically violates this IID assumption here. And finally, this is a very challenging data set where we have 3 blocks, each corresponding to an ethnicity. And furthermore, within each ethnicity, we have this very fine-grained structure. And so this is actually like a really hard problem. And to be fair, it's not that most data sets actually look this bad, but if you're interested in studying these methodologies, it's a very good one to work with. Right, so now let me walk you through what happens with a naive analysis through to sort of the class of models that I've worked on and that are the state of the art at the moment. So the most simple thing I could do is, first of all, let's imagine this is now a quantitative trait, so it's real valued, and let's assume for now it satisfies the properties of this model. We can abuse these models quite a bit, so that's not terribly important. But now if I just do linear regression, what I'm going to do is for each of my markers, which are called SNPs, I'm going to, one at a time, I'm going to put it into the model, and I'm going to evaluate basically how well that model fits the data. And then I'm going to pull it out, and then I'm going to get a difference, and that's going to give me a p-value. Then I'm going to do that over and over for each of my million markers. And if I do that on this real data set, is actually the data set I just showed you, the very difficult one, then this is what happens. And everything looks like it's significant, and in fact, nothing is significant. All right, so now I can start using this object here, this genetic similarity matrix. And so what can I do with it? So something that became quite famous from around 10 years ago from the Boston area, in fact, is called Eigenstract, is really a nice, clever application of principal components analysis. And so what you do is you select some number of principal components. You can roughly just use 5 to 10, or you can start using sort of more formal procedures to select them. And you essentially project each person from this very high dimensional space into just a few dimensions, and then you add this as a covariate to the model. And if you do that, you can capture structure that obeys this low rank property that principal components gives you. And so, but why should we think that this works? Well, so there are papers written on the theoretical basis for this in terms of population genetics, which I'm not going to go into, but this is, I think, a really cute figure which shows you the power of these things. So this is a genetic similarity matrix. And I'll tell you in a second exactly how we compute that. And then this is real data, and this was actually done several times. So if you take these people and you know their origin in Europe, and then you project each person down to the top 2 principal components, then you actually recapitulate like a warped map of Europe. And so it's not surprising that if this ethnicity is what's causing problems, and the genetics tags, because the genetics tags the ethnicity, and these principal components tag the ethnicity, then when I add these as covariates, it's one way we can correct for it. Right, so this works well, but sort of by definition in PCA, you're truncating the eigenspectrum, and so if there is signal in the rest of that matrix, then you can't deal with it. And so that's where these models called mixed models came in. And so you can think of these in very different ways, depending on whether you're a graphical models person or a frequent statistician and so forth. But I think the last line here makes it really, really easy. As long as you know linear regression, you can understand these models. And so, right, so what is this? If I cross out this term here, this is just linear regression written in vector form, so multivariate Gaussian. So normally you would see just a single number here for whether a person has cardiac disease or not. Now it's a single number for each person in the cohort. So there's n individuals here, and that means that this matrix here and this matrix here is an n by n matrix. And what you can see is if I cross this out, again, that's linear regression. And so that this encodes this IID assumption. And what the linear mixed model does is it puts this very, very rich structured noise term in to precisely kind of correct for this problem I've just told you about. Right, and so again, on that real data set, this is what happens with these 3 different models. This is uncorrected, where everything looks like it's associated with the disease. This is PCA corrected or eigenstrat corrected, and it's able to account for those 3 blocks, which are low rank structure from the 3 ethnicities, but it can't actually capture this very fine grained structure from the siblings and pedigrees. And the mixed model in this case can, and that's the blue model here. Right, so I spent quite a number of years working on these mixed models. And so I just basically showed you they work really well. So what exactly did I work on? So a few things. One is these are very, very expensive models, and to the point where even at the time I started working on this, there were data sets where you couldn't apply them. So people would spend millions of dollars collecting these samples, and then they couldn't apply a model, and they would actually have to, this is no joke, you would go to conferences, and there would be people publishing papers on ways to remove people from the data set so as not to have this problem. But of course we want everybody in the data set, and we just want the correct model. And my first foray here was making these models faster, and I'm going to give you a little bit of a flavor of how we did that. And then in the course of thinking about these models deeply to make them faster, we also started to think about how we could make them better. Right, so again, this is a big multivariate Gaussian model, and so why is it expensive? You don't need all the details here. All you need to know is that this matrix, right, this K matrix, it's n by n. If I have n people in my cohort, and n nowadays can easily be 1 million people, and I need to compute the likelihood of this model in order to get p-values, and I need to do it for every SNP. So this is like a lot of computation. And naively, you actually have to basically compute this over and over for every single SNP, like dozens of times. And every time you do that, you have to take a determinant, and you have to take an inverse of a matrix, which is n by n. And those are cubic operations, and you have to store it in memory, which is a quadratic space constraint. And that actually was too much for the data that was available already 5 years ago, and now definitely too much. Right, so we developed a few speedups. One is exact, and one is kind of an approximation or an improvement, depending how you view it. So let me just, again, give you a little flavor of how these things work. So remember that I said that the main computational bottleneck is this determinant and this inverse. And what I'm going to do is I'm going to replace basically having to do those over and over and over by a single operation that has the same expense. And it's going to be the spectral decomposition. So I'm going to take this matrix and just once up front, I'm going to compute this decomposition. And then I can shuffle around the terms in this model. So this is what I started with. And if I shuffle them around, I end up with this. And this is interesting for several reasons. So what it is you can think of U. Can think of U here as a rotation matrix. And what I'm doing is I'm rotating my trait vector, and I'm rotating my SNP or my marker and any other covariates that I have. And the most important part is that when I do that, what I'm left with is a diagonal term here. And what that means is essentially this equation looks in some sense a lot like linear regression. Linear regression is more than diagonal, it's actually spherical, but it doesn't really matter in terms of the expense of the computations. As soon as I have this diagonal, I get back down to the expense of linear regression, and things are, again, very, very fast. And so now the bottleneck is simply this one upfront computation here. And so that's what we sort of try to reduce next. Right, so how could we make that faster? So that's when we started getting into some approximations. So again, the key thing that I've boiled everything down to is this decomposition, the spectral decomposition of this matrix. And the way we actually compute this matrix, which so far I've glossed over, is actually an inner product distance. And so what you should think of is that basically as a data analysis person, someone has given me this cohort and say 1 million of these markers, these SNPs. And at the time that we were doing this work, people would naively take all of these markers to estimate this genetic similarity in this manner. And if you do that, then so n, let's say, is 30,000, but you have like 1 million of these markers, that means this is a full rank matrix. And that means that you have this cubic computational cost. However, as many of you may know here, if you're well-versed in linear algebra, I can get the decomposition of this guy by instead using the decomposition of this object here. You just square the eigenvalues. And why is that interesting? It's interesting because if I can somehow make the number of SNPs with which I estimate my genetic similarity less than the number of people in my data set, then it actually becomes linear in the number of people instead of cubic. And so actually it enables us to suddenly analyze data sets we literally couldn't analyze before. And the only consideration is, can we do that? Can I, instead of naively taking all these markers that I've been given, can I reduce it to some more, to a smaller set of markers? And so we played around with different ways to do that. In the first paper, we leveraged something called, well, it doesn't matter what it's called, but in the genome, basically locally along the genome, markers are correlated with each other due to the way that the chromosomes are reproduced. And so what that means is there's some redundancy in the genome. And so the closer you are together, the more correlated you are, and therefore I can essentially thin out SNPs and hope to retain almost as much information. And so that's basically what we do is we take, say, every 100th SNP or every 500th SNP, and then we use just those markers, and hoping that we'll still capture enough of the information, but get this computational savings. And so that worked well, and we could suddenly analyze data sets that other people could not analyze. But then we started to think about these models a bit more deeply and had a different idea. And so I don't need to go into this, but let's just say you can rewrite this model in a way such that the SNPs, or the markers you're using to estimate this genetic similarity, they're actually just covariates in my model. And so why is that interesting? It's interesting because as a data analysis person, if I'm, say, I have a sample size of, say, 30,000, someone gives me 1 million covariates, there's no way I'm just going to naively use those, right? I'm going to do some sort of feature selection or model selection. And that's not something people had been thinking about in this field. And coming from a machine learning background, is a pretty obvious thing to try. And so we thought, instead of just doing this thinning based on the redundancy, let's see if we can do something more clever where we achieve the gains in computation while actually also making the model better. And it turns out we could do that, and it works quite well. So, and we have software that goes with this that actually people all over the world are using right now. So I'm going to now do the rest. Are there any questions on that part? Okay. So I hope to have given you a lot of intuition about what the problem was there and the kinds of analyses we do. And so there's this guy, James Zhao, was a student at Harvard, and he approached me for an internship, and he said, I've been following your work, it's very interesting, and I've seen a problem in a related area that seems it's different, but it's analogous, and I think maybe we could use these models, like, do you want to work on this together? And so that's what we did. And this has to do with the epigenome now. These are these markers, I said, that affect the shape of the DNA. And so there's something, I'm going to focus on a particular one called DNA methylation. But the key thing to know about epigenetics is that it's cell type specific. So far what I've been telling you about is just sort of regular genetics. And if I take a sample from my hair or my cheek or something like this, it doesn't really matter. Effectively, I get the same measurements. But when I go to something like the epigenome, then like the cheek and the hair, like these are wildly different things. And so if you're trying to understand how epigenetics plays a role in disease, and you have some sample that's a heterogeneous sample of cell types, so this is like a solid tumor, this is a big jumble of cells, and the epigenetics in there is different for everything. And it turns out that there's a direct analogy between the cell type heterogeneity and the sort of population structure that I told you about in the previous problem. And in fact, you can draw these kinds of plots again. So now instead of checking each marker in the genome, I'm checking each methylation sort of position in the genome and doing something very analogous. And you'll see that this plot actually looks much, much worse than anything I showed you in the genetics space. And in fact, when I started giving this talk, I had people who do this for a living. They generate these data, and they would come up to me and they'd say, oh my God, this is what my data looks like. I thought I'd done everything wrong. Like, can you help me? And so people started developing models to fix this problem, and this is what they did is this reference-based approach. And so what they do is they basically have to know which cell types are present in the sample, and then they need to purify out those cell types and essentially get like a reference profile for every possible cell type. And now then for each person, you need some deconvolution process that effectively projects you onto each of those reference templates. So you now have an estimate of what proportion of each cell type you have in each person's sample. And then you can add those as covariates to the model, and it completely fixes the problem. So that's great. So where is their work? So there's a lot of problems with this. It depends on knowing the cell types, depends on isolating them, and all kinds of things that actually can go wrong. And people have actually only done this for blood, but people are very interested in doing this for solid tumors like breast cancer and all kinds of other things. And so our goal was to basically use these methods like mixed models and principal components analysis to do this in a totally reference-free way, thereby sort of getting rid of all those potentially problematic things. And in fact, that's what we did. And you can see that the distributions look very similar. Moreover, if you drill down into those distributions, you'll find that there are 5 significant markers, and this is from a paper earlier this year on rheumatoid arthritis, and they validated these. So we know that these are very reasonable hits, and these are the things we kind of should be finding. And what you see is that our approach against this reference-based approach yields essentially the same answer, except we bypass all those sort of headaches that they had to deal with. And moreover, we actually get the same effect size to like a precision of 2. Which is pretty amazing. And then this plot is quite fascinating, because what this shows you is that if you take a good method, like our method or the sort of bronze standard method, which performs similarly, versus an uncorrected method, where you ignore this problem, what you see is that those top 5 hits that you should be getting out, they're basically stuck behind like thousands and thousands of markers. So in other words, if you don't do the right thing here, you've just completely thrown out your data entirely. Like you may as well not have done the experiment. So this is very, very important to think about and to do. Right, so now I'm going to go into the last half of the talk about gene editing. Any questions yet?
So,
yeah.
So going back to the first part, you were talking about SNPs as a particular type. That's a particular type of genetic cause,
Yeah.
but of course not the only kind. Do any of your methods carry over to thinking about multi-site mutations that would cause then phenotype
Yeah,
of disease?
absolutely. You can generalize almost all of those concepts. So people often test sets of markers within a gene to try and get higher power, which you can sometimes do, or across pathways. So there's many flavors of this, which we have published on. Just for simplicity, I didn't touch on that here. Yeah. Right. Okay. So probably most people here have in some way heard of gene editing, and gene editing can mean kind of 2 different things. It can mean that I edit a nucleotide, say from a T to an A. It can also mean just that I want to knock out the gene. You might want to do that for therapeutics. You might want to do that to probe a biological system. But the point is that there's one common task here, which is I need to go in and very precisely cut the genome at a particular place and not cut it elsewhere. Right, so people have been pursuing this goal for many decades, and in the last 5 to 10 years, this one called CRISPR has come along that's really had a major impact on society. It's going to get a Nobel Prize. There's the patent wars, which many of us have been following. And why is it so exciting? Well, it's much easier to engineer. It's much, much cheaper, and it's more precise. And so as a consequence of that, it's been like getting lots of media attention. So starting, in the primary scientific journals through to the popular press like CNN and the New York Times. And then I don't know, did anyone see this cover article from 2015? So this is all about CRISPR. And according to Wired, it's going to be no more hunger, no pollution, no disease, and the end of life as we know it. And so after that was published, there was a bit of backlash on Twitter. And there's many more. I used to change these in between talks, but I think it's reached a steady state now. But more seriously, this is really an exciting technology, and there's already very promising results here for translational medicine, and I've listed a few here. I think I alluded to some of them at the beginning of my talk. So altering like a receptor so that you don't acquire HIV, or correcting a defect to prevent you from getting cystic fibrosis and things like this. So there's if I tried to update this, literally I would have to update it every day with these kinds of experiments. But it's not quite ready for prime time. Those were like in model organisms. And so why isn't it ready? So these are the two main reasons it's not ready. And this is going to be now in reference to this gene knockout, where I want to just stop the gene from functioning. So imagine I want to stop this gene from functioning. I can target it with this CRISPR system here, 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 here. There's a few constraints, so I can't target it everywhere, but I can target it very many places. And it turns out that some of those will work well, and some of those won't work well. And so I'm some lab somewhere in the world, and I want to know where to target it. Well, right now, before our work, you didn't have a great idea. And so that's how we can use machine learning to help us in there. And the flip side of that problem is, imagine I've decided to cut that gene right here. Then the question is, am I elsewhere in the genome? Am I going to actually also accidentally going to cut there? Because if I am, and this is therapeutic or something, that's like a disaster. And even if I'm just probing a biological system, it's certainly not ideal. It's adding a lot of noise to the system. And so, this is a rapidly moving area of molecular biology. Like every day, practically, there's some new, very big paper on improving the molecular biology to address these problems and also to generalize the system for other uses. But the reason I'm here talking to you today is that we decided to use machine learning as well. And obviously, these, can be synergistic with each other. So, but first, I want to tell you a little history of CRISPR because I think it's really fascinating. So for many decades, people saw these weird patterns in the genome of bacteria, and they didn't really know what they were. And then it was actually a yogurt company, a scientist at a yogurt company, who figured out what was going on. And only in 2007, after decades, and these little weird patterns are actually what is now called CRISPR, which stands for Clustered Regularly Interspaced Palindromic Repeats. And so it turns out it had to do with a naturally occurring immune system in bacteria. And which I'll tell you about in just a second. But very shortly after, you can see just 5 years later, people already figured out how to kind of co-opt the system in order to do general purpose gene editing. And it's just the field's been exploding ever since then. All right, so how does it work in its native system? So here's a little cartoon. This is not real data of a bacteria. And there's a little invading virus. And so if the bacteria survives this invasion, what it does is it essentially, much like yours and my adaptive immune system does, makes a little copy or a little sort of memory of this virus. And in the case of CRISPR and bacteria, it stores it in its own genome. And so as to sort of tease apart that these are these special markers, it uses little demarcations here. And these are the actual CRISPRs that sort of separate these viral memories. I forgot the second half of the story, right? So it stores them, and now if it gets infected again, basically it's primed to recognize this virus, and it has these innate sort of scissors in there, and it basically immediately can hone in and go to the right part, cut the virus, and thereby disable it. And so there's this, I keep going back and forth. So there's clearly this mechanism that enables you to go to a precise part of this viral genome and cut it. And so people started to think, maybe I can use this for gene editing. And that's exactly what they did. It turns out you can actually just take these parts of the system, kind of throw it into almost any organism and get it to work. Except there's one difference, which is we don't want to wait till something's infected with this sort of virus template, right? We want to tell it where it's going to deploy these scissors. So we're going to synthetically create what used to be a sort of viral memory. And in the lingo of CRISPR, that's called a guide RNA. And so I'm going to tell you about machine learning and guide design. That is like creating guides that do like good on-target and reduce off-target activity. Right, so for the first one, this is sort of a standard supervised machine learning setup here. And so what is, I have these inputs, which is the guide, which is basically 30 nucleotides long, so ACTG. And then we have a very wonderful collaborator at the Broad Institute who measures these things in his lab. And for 17 genes, he systematically measured everywhere that you could deploy the system and how effective it is. And actually, I don't have time to go into this, but that's a very, very difficult thing to do, and you can't do it for most genes, actually. So even if you had the time and money to do this for everything, you actually can't do it technology-wise even. And so our goal, of course, for predictive machine learning is to learn this function that takes us to the guide and then how well it managed to knock out the gene. Right, okay. Right. I'm not going to go into the details of that model, but some interesting things that emerge are the kinds of features that are important here. And in case you're thinking like, oh, did you use deep neural networks who learned their own features? No, there's not anywhere near enough data to use deep neural networks here. So we need to go old style. And John, who's the biologist who works with us, is a bit of an oracle for us sometimes on features. We can use the rich history of computational biology to help us and sort of a bit of trial and error. But one interesting thing that comes out is the thermodynamics of the guide come into play. And why is that? So you can see in this video here, these are the scissors, the Cas9, and they actually have to go in and they have to pull apart the double-stranded DNA, which obviously takes energy. And the amount of energy it takes depends on different, what the nucleotides are, the strength of the bonds and things like this. And therefore, it's not surprising that, in fact, the thermodynamics do play a big role. And if you look at the crystallography that's been done for this system, you can even further hypothesize sort of subsections based on the dynamics as it comes into bind that might be important. And it turns out these sort of subparts of the DNA you want to model separately. Right. So so people had sort of dabbled a little bit in machine learning here, but to be honest, not very rigorously, not very carefully. And we spent a long time just saying, we know this is important to the world, we know people will use this. And so in the end, this is the model we finally deployed, which we're actually just updating right now. And you can see on several different data sets that it's the state of the art compared to what was out there before. And the sort of models spanning performance in between are actually just our own models. And it's been very nice to hear after publishing this that 2 other groups have since actually independently validated this now as the state of the art. And it's actually been adopted by 2 startups, and people download this, and it's getting a lot of use. In fact, whenever I go talk to biology departments, like all over the world, in fact, usually someone comes up to me and says that they're using this. So that's the on-target problem. And so now I'm going to tell you about the off-target problem, and that's actually a much harder problem for multiple reasons. And so the main thing to think about why it's harder is that I'm looking for accidents, right?
What is a model?
Oh, that model is just boosted regression trees, yeah. Right, and so the modeling here is a bit more interesting, but we really have our hands tied because of how little data there is right now. And so, right, so the off-target problem is I've chosen this guide, and basically I want to estimate everywhere else in the genome how likely it is to cut. And moreover, I actually also want a single aggregate number, because the way people use these kinds of models is there's someone in a lab somewhere down the street, and they say, I want to knock out this gene, and I know that CRISPR lets me try these like 700 guides, and I don't know which one to use. And can you tell me the on-target activity and the overall off-target activity for each of these guides? So it's not even quite enough to just know each of these individual numbers. I need some way to aggregate them. But right, so why is it hard? It's hard because I have to scan all over the rest of the genome just for this one guide. The genome's 3 billion base pairs, so that's already like a hefty search. And then additionally, in terms of the machine learning, I get this sort of combinatoric explosion going on, because now, whereas before the guide was meant to be, or was exactly complementary to the target by design, now I'm looking for accidents, and I need to consider what happens if there are mismatches between these things, and whether it still might have activity. And in fact, you can go up to like 7 mismatches and still get activity. So you need to be really, really careful. And so we can break down this problem into three sort of sub-problems. And the first one is we need to basically, for a given guide, I'm going to scan the genome and develop a short list of candidates that I'll then use machine learning on. And I can't do everything because it's just too expensive. And I'll tell you, we have a simple heuristic for this. We could use machine learning, but we haven't yet done that. And now once I have this short list of candidates that might be a problem for this one guide, then I'm going to use machine learning to score each of those guide target pairs. And then finally, I'm going to aggregate these all together using machine learning. And the second step here also uses machine learning. Right, so first of all, you can see that as I tolerate more and more mismatches, I have to have a much, much larger short list from the genome. And this just gets really unwieldy, but we have to deal with it because, in fact, up to quite high, it does still active. And here this says up to 3 mismatches, but it's sort of something a bit more complicated that lets you dive down further than that. But so in other words, there's some heuristic where we just say we cut it off here, and that's our short list. And now we go back and we do some more predictive machine learning. And here the problem is a bit more difficult. So previously, there was only a guide here, and that's because the guide and the target were by design complementary. But now I need to tolerate these mismatches. And so now I need to have my function take both the guide and the target and then predict this off-target activity. And so you get, again, this combinatoric explosion. We have very, very little data here, like in the thousands. So it's really, really hard to do this. And so really the only way we managed to do this was to make some sort of assumptions of independence in a weird way and then to correct for them as best as we could. And so we have some data that has only single mismatches. For a single gene, it's been exhaustively tried how well every single mismatch, what the activity is like. And so we can build a model for single mismatches from that data. There's no combinatoric explosion here, so I can get a reasonable amount of data and do a pretty good job. Now I have some example where I have more than one mismatch. So how can I sort of bootstrap myself up to getting some reasonable thing? So I can make, roughly speaking, I can use the single mismatch model to get sort of a score for each of these sort of now pseudo examples. And then I can make some assumption of independence and kind of multiply them together. And that actually doesn't work that badly. But what we decided to do is add another layer of machine learning on top of that, just very, very simple L1 regression, in fact. It has to be dead simple because there's so little multi-mismatch data that you just can't use a richer model. And it turns out that when you do that, you still get, it does help that last layer. But one thing we spent a lot of time on, more than any other project I've worked on, was actually how to evaluate this. So, there's standard things, there's ROC, and there's correlations, and blah, blah. And but no matter how you look at this data, nothing quite seemed like the right way. And one of the reasons for that is there's an inherent asymmetry in the errors you can make. So think of these two examples. Imagine that there's somewhere in the genome where it truly is very, very active and it disrupts the genome, so where you don't want it to. If my model tells you there that it's not going to disrupt it, then that's like a really grievous error, right? If it's therapeutic use or whatever, it's going to do something dramatically bad. The flip side is if my model makes the other kind of error, so my model tells you that it's got very bad off-target activity here, but in fact it doesn't, well then I just don't use it. So I limit my choice, but not something sort of really terrible doesn't happen. So I know there's this asymmetry, but I don't actually know exactly where I sit between those, how I want to trade them off. And so what we ended up doing is sort of doing the following. On the right-hand side here, these are 4 models. The red one is ours. And we use just a non-weighted Spearman correlation, and you can see that we do very well. And then what we said is, let's do the other extreme here, where we do a weighted Spearman, where we weight it by how much it was knocked out. So what that means, and this is sort of an extreme version of this asymmetry, because here, if this guide target pair wasn't active, it falls out of the metric altogether. And then what you can do is you can kind of turn that knob in a smooth way and trace out this metric. And you'll see that, in fact, here, each method dominates the other along the whole spectrum. And there's not some theoretical reason for this. It just happens to be, which is lucky for us, because what it means is then, it doesn't matter which knob setting we want, which might actually vary for different use cases, because systematically across the board, our model is still the best. Right, so now I've told you how, for a given guide, I can estimate at every position in the genome that I've shortlisted how likely it is to disrupt the genome there. But now I want a single measure. So how can I do that? I need to take this sort of one guide that I've decided to use. I shortlist all of its potential off-targets into a long list, and then I apply the model that I just told you about, and it gives me some number for that guide target pair. Now I need to aggregate all of these things into a single number. And again, like, we don't have a lot of data here either, so we can't do super fancy things. But ultimately, we featureize this distribution in terms of moments and min-max and whether it lies in a gene and things like this. And then we, again, apply machine learning. And you might ask, well, how can I even do machine learning? Like, what is this aggregate signal exactly? So we can't quite get an aggregate signal, but we can get our hands on it in this particular way that I'll show you here. It's called a cell viability assay. So it measures whether a cell survives. Then what we do is we take CRISPR and we look only at what are called non-essential genes. So non-essential genes are genes that you can stop from functioning and the cell still lives. So if I take CRISPR and I try to target its on-target activity only for non-essential genes, if the cell dies, it's because there was off-target activity, by definition. And so now I have this proxy of sort of the aggregate off-target activity as to by how well the cell lived. And there's actually a reasonable amount of data like this right now. And so if you do that, you'll see, in fact, that the Spearman correlation here is not very high. It's only 0.1. So we have a long way to go, and I think with more data that will become much higher. But importantly, this was the competition. And in fact, this is the most widely used website in the world, from MIT and the famous CRISPR people. And they're brilliant molecular biologists, but they've never really tested their model. They just kind of make it up and then put it out there. And so we've clearly made a good advance here. Anyway, so now that part's in revision. The first part is published, and we're putting it all together now because people really, really use this, like, all over the place. And they would like to use it quickly. And so those computations take a really long time for the off-target because there's all of this search and these kinds of things. And so what we did is for the human genome, we took every place that has a gene, which are the places usually people want to, because you want to knock out the gene. And for everyone, we pre-computed all of these numbers, including all of the off-target, and we pre-populated it and put it there, and people can kind of drill down. And so to populate this, just to give you a sense of the computational cost on very simple models here, like not deep neural networks, we ran 17,000 cores for 3 weeks straight. And actually, we have to redo it now, because we didn't put something in that we want. Right. Anyway, so that's the end of CRISPR, and I'll just thank my collaborators now. So the GWAS stuff was with Christoph and David, who are actually at Human Longevity Institute right now, and James, who's faculty at Stanford. And the CRISPR stuff is my colleague here at MSR New England, Nicolo, and John, who's the hardcore CRISPR biologist at the Broad Institute just across the street. Thank you very much.